Hi, so today I'll be talking about heart disease, epigenetics, and metals, and studies that have been done in the Strong Heart Study. So a lot of my work has focused on exposure to arsenic. So I'm going to start by talking about arsenic, but also touch on some other metals in this presentation. Although we've made a lot of progress to decrease exposure to arsenic, particularly through drinking water, arsenic exposure remains a major public health concern. This map shows the probability of groundwater arsenic concentrations exceeding the US EPA maximum contaminant level of 10 micrograms per liter. And in the orange and red sections of the map here are clusters of higher exposure to arsenic. So you can see areas of the South and Southwest and some parts of the Northeast in Maine. And many of these areas with high arsenic concentrations also correspond to areas with a high reliance on domestic wells for drinking water, which aren't regulated by the EPA. And the map here shows higher populations that rely on domestic wells for drinking water, shown in darker areas of green. A couple of years ago, this issue was highlighted by the New York Times, which told the story of a mobile home park in an agricultural area of California. And residents here were exposed to very elevated levels of arsenic in drinking water. This resulted in a range of sicknesses, including rashes and kidney failure. The picture here shows a boy with a rash on his back, and then there's a picture of a woman showing here that she had collected, that she had lost. So contaminated water can be a major issue for mobile home parks where people often own their homes, but not the land that they sit on. So they don't necessarily have control over where their water comes from. And before I get too far, I just want to take a couple of minutes and talk about my own background and how I became interested in this area of research. So this is where I grew up in the Mojave Desert. And I first began thinking about how health is related to the environment back in high school. Around this time, the EPA lowered the maximum contaminant level for arsenic from 50 to 10 micrograms per liter. And there was some pushback from community members when it came to installing water filtration plants, partly because it was so expensive to do and there aren't a lot of resources in this area. And zooming in a little bit, this is where my family lived in a mobile home in the bottom of the picture here. In the middle of the picture, you can see a small mobile home park, and right across the street is a really large auto salvage yard. So this is another source of contaminants and groundwater contamination. And then across the big road at the top here, this is where the Navy base starts, where they did weapons testing. And there's actually a cancer cluster associated with this base. So you can see how exposures to multiple toxicants and carcinogens can start to layer on. And I mentioned a few of the health effects related to arsenic, but we know that arsenic is a human carcinogen and toxicant, and it has adverse health effects that span across the life course and affect most organ systems. In addition to diseases like cancer, arsenic can be related to increased risk of chronic diseases, including cardiovascular disease and diabetes. So a lot of these arsenic-related health risks persist decades after exposure has ended or been reduced. These plots are from a study in northern Chile, and they show that the risks of lung and bladder cancer in adulthood are increased after arsenic exposure, and particularly high when the arsenic exposure was limited to early life periods or even exposure prenatally. So this really long latency period suggests that epigenetics may be involved in linking arsenic exposure to health. And in a couple of slides, I'll talk about what I mean in particular by the term epigenetics. But these chronic diseases that I'm talking about all have greater effects among minority populations compared to whites, particularly among indigenous communities. The plot on the left here shows the average age of death from a number of different chronic diseases, including heart disease and lung cancer, CPOD, diabetes, and stroke. And the bars show the average age of death for white women and white men, whereas the lines show the average age of death for American Indian and Alaska Native women and men. So you can see that the average age for indigenous communities, average age of death from these chronic diseases for indigenous communities is a lot lower than those for whites. On the right here, this shows the proportion of people who have diabetes by race and ethnicity. And the greatest proportion is among American Indians, even greater than that among Blacks, Hispanics, or Asian populations. So that's really important. One of the reasons that the Strong Heart Study was developed and really focusing on these indigenous communities, which often overlap with areas of high exposure to environmental contaminants, including arsenic. And of course, arsenic contamination in groundwater isn't the only concern. A lot of these exposures also come from mining activities. The map here on the left shows the location of active or abandoned gold mining and how many of these are in close proximity to American Indian reservations. In addition, uranium mining can contaminate water, and the plot here on the right shows contamination of uranium in public water systems, which is also a major concern for areas of North and South Dakota. So you already got a good overview of the Strong Heart Study earlier this morning, but I just want to emphasize the strengths of the study. The Strong Heart Study started over 30 years ago to study cardiovascular disease among American Indians. And it enrolled participants from 13 tribes and communities in Phoenix, Arizona, the southwest area of Oklahoma, and as well as North and South Dakota. And this is the largest epidemiological study of American Indians ever taken, and enrolled about 4,500 participants. And then in the late 1990s and early 2000s, the Strong Heart Study was expanded to the family study and enrolled another 3,700 participants from 94 families of the original study. And we really have a wealth of data from the Strong Heart Study with data collected over the course of decades. So we have data that was generated both from blood samples and urine samples. And using blood samples, we've been able to measure DNA methylation, which is one epigenetic marker, as well as other omics, for example, the metabolome, RNA expression, and genetics. And then in urine samples, we've been able to measure metals, including arsenic, over the course of decades. So being able to look at changes in exposure over time. This is a slide that was created by Alison Kupsko, so I just want to give her credit, but I think this is a really great illustration of all of the wealth of information and culmination over the Strong Heart Study that has generated information to support links between metal exposures and chronic diseases. In addition to arsenic, we've also found links between zinc, cadmium, and uranium and the incidence of chronic diseases. So as you know from the title of this presentation, I'll be focusing on epigenetics, but what do we really mean by this term? You might have heard it in the news or maybe on a TikTok video, so I'll try to explain more specifically what I mean. I'm going to use an example to start with. This is a piece of paper with what looks like just a bunch of random words on it. This is kind of similar to DNA in a cell. So this is a picture of a chromosome, which contains DNA and a gene. So this genetic sequence gives information to a cell on how to form a cell and what genes to express. So by highlighting certain words on this paper, it suddenly makes a lot more sense. I'll read the words that are highlighted in green. The environment is where we all meet, where we all have a mutual interest, is the one thing that all of us share. And then if I highlight another set of words and read that, the blue words, whisk flour, sugar, baking powder, baking soda, and salt together in a bowl. So now this is a sentence from a recipe. So two completely different meanings extracted from the same set of words. So we can think of DNA as the same way, and epigenetics is a way of highlighting the parts of DNA that are important. So here we highlighted the green sequences of DNA, and this contains information on how to form a neuron cell, for example. And then if we highlight a different set of genes, the blue genes give information on how to make a blood cell. So what happens if maybe a friend or a child comes along and highlights some extra words on this page? And I'll read the green words again. The training environment is where we all meet, where we all have a mutual interest. Is the salt one thing that all of us share? So it kind of makes sense, but has a couple of errors in it. So the same thing can happen to epigenetics as it is exposed, as an organism is exposed to environmental toxicants. The epigenetic patterns on our DNA can change and lead to adverse health outcomes. So for a more formal definition, epigenetics refers to modifiable information above or beyond the genome. So that's where the epi part of the word comes from. It's also a form of cellular memory that's stable, so passed on between cell divisions. Today I'll be focusing on DNA methylation. This refers to a methyl group, the addition of a methyl group to a cytosine residue, as shown in the little orange circle here. This is usually at a cytosine followed by a guanine. These are just the names for different bases that make up the sequence of DNA. So this combination is known as a CPG site. So if I say CPG site, that just refers to a position that can be methylated or have an epigenetic modification on it. And these changes are involved in controlling gene expression, and they're also important for maintaining genomic stability. So we're interested in changes that are more subtle than just an on and off switch. We're really interested in variation in the percentage of DNA fragments that are methylated at a specific site. So in the picture here, these are all the same location on a string of DNA, and 8 of them are methylated, have the little orange circle on them. So we refer to this as 80% methylated. So I'm really interested in epigenetics as this bridge linking the environment to health. And we can measure DNA methylation and epigenetics to look at how epigenetic dysregulation is involved in molecular pathways linking environmental exposure to health. We can also use epigenetics as a biomarker of past exposure and future disease risk. And by biomarker, I just mean an indicator of biological processes, so not something that's necessarily mechanistically involved. And we can also look at how changes in DNA methylation are related to other omics layers, for example, changes in metabolomics, and look at how that is involved in larger pathways and physiological changes. So I'm going to talk about some studies in particular that have been done in the Strong Heart study. The first study, we were really interested in being able to look at DNA methylation as a way of measuring biological pathways that are changing as a result of exposure to arsenic. So this is a screenshot of the paper that's been published on this study. But we use data from the first visit in the Strong Heart study, so both DNA methylation data and arsenic measured in urine. And we were able to find that DNA methylation at 20 sites were associated with arsenic exposure measured in urine. We also identified one differentially methylated region, which is shown in the plot here. And what I mean by this is a cluster of CPG sites that had higher methylation levels with higher exposure to arsenic. We also found that genes that had differential methylation were related to oxidative stress, tumor development, and glucose metabolism. So all pathways that are important for chronic disease development. And when we wrote this paper, we concluded by saying that further research is needed in more diverse populations and to investigate whether arsenic-related DNA methylation may serve as biomarkers of disease development. So that's exactly what we did in the next study I'll talk about. This was led by Artha D'Amico Rioso, and it's a pretty complicated study, so she did a great job of tying everything together. So first we looked at this association between arsenic exposure and changes in DNA methylation. And then looking at how these changes in DNA methylation were related to cardiovascular disease. And this used data from the Strong Heart study, as well as 3 other cohorts from diverse populations. We also used data from an animal study looking at how arsenic exposure is affecting DNA methylation in mice. And this is important because we have a really controlled environment to study changes associated with this environmental exposure. We found that DNA methylation was a potential mediator between arsenic and cardiovascular disease incidence and mortality. We also found that arsenic-associated sites were similarly associated with incidence cardiovascular disease in 3 diverse cohorts. And in the mouse model, we found differential methylation was also associated with arsenic. So by combining all of this evidence from different types of analyses, we were really able to strengthen the causal argument, strengthen the argument that there is a causal link between arsenic exposure, DNA methylation, and cardiovascular disease. So as I mentioned, we can use epigenetics and DNA methylation as a biomarker as well. And as a biomarker of previous exposure. So sometimes it's often hard to measure past exposure. We may not have a urine sample, for example, to measure exposure to metal or a blood sample. Also, if we ask participants, how much arsenic were you exposed to in the past? Most people have no idea. Also, certain types of exposure, like exposure to trauma or illegal substance use, might be difficult to collect. So we can estimate these exposures by DNA methylation levels. And we used an existing biomarker that was developed to estimate lead exposure. And we applied this to the Strong Heart study. And this was a study that was led by Will Lieberman-Krebin. And he estimated lead exposure based on DNA methylation levels and looked at how this was associated with cardiovascular disease, both incidence and mortality. And he found that greater estimated levels of lead exposure were associated with increased cardiovascular disease mortality. And we can also use epigenetics to estimate health status. So DNA methylation at specific CPG sites changes predictably as we age. So we can use this to estimate the age of a sample. This can be interesting, but not necessarily super useful in epidemiological studies where we usually know the age of participants. So what we're really interested in is the difference between epigenetic age and chronological age. So on this plot, chronological age is plotted on the x-axis or the horizontal axis here. Epigenetic age is plotted on the y-axis or the vertical axis. This arrow is pointing to a specific person. So this participant, their chronological age was 88 years old, but their epigenetic age was only 77 years old. So this person is 11 years biologically younger than they are chronologically. So everyone falling in this blue area of the plot has negative, what we call epigenetic age acceleration. So they're biologically younger than they are chronologically. Everyone in the orange area has positive epigenetic age acceleration, so they're biologically older. And we can think of aging as these trajectories. So we'd like to be able to shift these trajectories upward to be able to extend functional longevity. So epigenetic age acceleration has been associated with adverse health outcomes, including neurodegenerative diseases, cancer morbidity, and mortality. So all chronic diseases that have relevance for environmental exposures and have been linked to exposures, metal exposures such as arsenic. So in this final study I'll talk about, we were really interested in looking at how multiple metal exposures were associated with epigenetic age acceleration. And this was a project done by Kelly Boyer. So she analyzed the associations between individual metals and epigenetic age acceleration and found that higher exposure to cadmium, so people in the 25th percentile and above, had greater epigenetic age acceleration or biological age measured by 3 epigenetic clocks. We also found associations between greater zinc exposure and epigenetic age acceleration, although it wasn't necessarily as much of a linear association. So we also looked at the effect of metal mixtures and compared mixtures of essential and non-essential metals or toxic metals. So we found that higher levels of essential metals, so metals that you need for health and functioning, were associated with lower epigenetic age acceleration. But higher non-essential or toxic metals were associated with greater epigenetic age acceleration. So just to summarize, I talked about a number of studies, but I want to highlight how all this data from the Strong Heart study has really helped to strengthen evidence linking metal exposures to health. So I talked about a study showing that arsenic is associated with changes in epigenetics and subsequently the onset of cardiovascular disease. So a potential mechanism linking environmental exposures to health. And I also talked about a study that allowed us to examine the impact of lead exposure, even though we didn't have measured levels of lead exposure in the Strong Heart study. But we were able to use this epigenetic biomarker of lead to link lead exposure to health. And we were also able to estimate long-term impacts on aging and health by using, again, one of these epigenetic biomarkers and looking at how metal exposures were affecting biological aging. And oftentimes these are framed as higher levels of environmental exposure are associated with adverse health outcomes, but we can also use these epigenetic aging biomarkers to indicate potential improvements of health and decreases in biological aging. So I just want to thank everyone that was involved in this research. The Superfund Research Project at Columbia, Anna Navas-Asian, who leads the project, as well as Alison Kupsko, who made a lot of the slides that I presented today. I want to thank Missouri Breaks Research Institute, which is really important for conducting all this research, as well as all of the participants involved in the Strong Heart study. We couldn't do any of this work without them. And I will just wrap up here.